Now we will start with the cell signaling, and the first KPI, describe the basic components of the cell communication. We should know like what do we mean by the communication. So of course for a communication we should have a stimulus, then we should have a receptor cell, and then there is transduction, and then in the end we get a response. So these are the parts of the cell signaling, where the stimulus can be like a signaling molecule that can also be called as a ligand that bind to receptor cells on specific receptors, and these receptors can be either on cell membrane, they can be either on the cell membrane or inside the cell, depending on the type of the ligand, if it is a hydrophobic or hydrophilic. And transduction is like when the gene turns on or off. So this is related to switching on or switching off the gene, and response accordingly. 4 students were asked to list the basic components of the cell communication, and their answers are shown in the table. Which student gave the correct answer? So we need to choose the same thing as we discussed above. Number 1, cell signaling molecule, a receptor, and target cell. As we can see what we wrote over here, signaling cell is like the receptor cell. Okay, signaling molecule is like the stimulus. Receptor, of course, the receptor cell have receptors, and then the target cell, of course, the receptors are present on the target cell. Yes, this is correct for so far. Signaling molecule, signaling cell, signaling molecule, transport protein. No, this is wrong over here. Nerve cell is wrong over here. Nerve cell, calcium ions, ducts, so all these are wrong. So only one. So you can go with A. Which statement about the receptors is true? So receptors are like, those proteins which are present on the target cell. So those are called as the receptors, and they are just like enzyme substrate reactions. So receptors are very specific. Receptors are very specific to who? To the signaling molecule, that's called as the ligand. Okay, so that's what we have to see. Most receptors have no connection. No, that's wrong. Receptors are found only on the surface. No, as I told you, receptors can be either on the surface or inside the cell. So this is wrong also. A receptor is specific to ligand. Yes, most receptors can bind many different. No, they are very specific. So this is wrong. Which statement about the receptors and the ligand binding is true? Is same as enzyme substrate complex. So remember in this one, so both must be protein in nature. That's not true. Both need to be present at higher amount. That's not true. It is irreversible. That's not true. It is reversible. Yes, that's true. Because after an enzyme substrate complex, enzyme is reused and substrate converts into the products. Which happens after a successful interaction between receptor and ligand? Of course, transduction will take place. So transduction, and then the response will come. So you can see over here, whenever there is a stimulus, it binds to the receptors on the receptor cell, transduction takes place, and we get a response. So this is what should happen after a successful binding. Signaling cell disintegrate? No. Signaling cell release more ligand? No. Target cells produce more receptors? No. Receptors are already present over there, and target cells respond to this interaction? Yes. Okay, we move to the next KPI. Describe using visuals how the cell communication over various long distance signaling, that is the endocrine, short distance signaling, that is the paracrine and autocrine, contact dependent signaling, that is the direct contact, gap junction, and plasmodesmata. So we use these below diagrams to answer questions 33 and 33, 34, 35. So this is like 33 till 35. So diagram number 1, it's, you can see over here, is a direct contact, right? It's a direct contact between the cell. This one is showing like electrical signaling, releasing molecule X, and then it's going to the target cell. So this is like kind of synapses. Okay, and synapses is paracrine. So remember, it is paracrine when the two cells are in very close vicinity. and then signaling molecule is transported through the blood cells and going to the target cell. Z is the target cell. So this is endocrine, long distance. Okay, which of the following is or are considered as content dependent? We already said it is one only. Which of the following? So look, this is easy when you analyze the diagram and then go to the question. Which of the following is considered as long distance? We know that 3 only. Which of the following is considered as the paracrine? The option is not given over here, 1 only, so it should be 2 only. Okay, so let's make it 2 only, then you can choose this, otherwise no correct answer is given. Which of the following, which type of the cellular communication is shown in the diagram below? Cellular communication. So of course, these are the cells which are like signaling molecules. So this is a ligand. Okay, then you need the blood, and then you transport to the target cell, and you can also see like the signaling receptors are present inside the target cell. Okay, they are inside the target cell. This is what else you have to see. But based on that, if we look, this is like hydrophobic ligand, while you have to look into it, blood is involved over here, so it's a long distance, right? So it's related to the long distance. And if you go back to the KPI, the long distance signaling is endocrine, so you can just simply go down again and choose the answer, endocrine signaling. Describe using representations of the 3 stages of the cell signaling. So reception is the first stage, transduction is the second stage, and response is the third stage. So a researcher is studying how a hormone triggers a response in the target cells. The process involves 3 stages. 4 students were asked to give an example of each stage from the muscle cell. Which student gave the most correct answer? Okay, so you should know reception is the binding of the ligand to the receptor. Then transduction will happen, that is switching on or switching off the gene related to the cascade reactions and protein synthesis. And then the response is like how the further reaction happens. Adrenaline, which is a ligand, it binds to its receptor cell on yes, that's correct one. You can also decide by only reading the first options or only reading the second. So let's read only first. The receptor hormone complex directly activates the enzyme. No, they don't. Okay, let's say yes, but we can see the next also. Let's go with the first one. The signal is relayed through a cascade. Yes, that's correct. And glycogen is broken down into glucose. Yes, that's correct. So all 3 are correct. So I'm gonna go with option 1 as a correct answer. Use the diagram below which to answer 38. This is the same question like the 3 steps. This is reception. Okay, this is the reception. This is transduction, a cascade transduction, cascade reactions that result in the formation of the protein or what, and then this is the response, like how the cell is responding. So these are the 3 stages. So you can simply, I already wrote them up, so you can say the 3 stages, reception, transduction, and response, and learn the description. For example, what is reception? When the receptor molecule when the signaling molecule or the ligand, it binds to its receptor which is present in the cell membrane, and the receptor become activated. So it's showing you the ligand receptor reaction. Step number 2 is transduction, where the relay proteins, you can see over here, many relay proteins are the cascade reactions of the proteins get activated in series of reaction inside the cell. And the last one over here, the response, you can simply say that the packaging of glucose molecule is taking place over here. So glucose molecules are being packaged and transported across the plasma membrane. Complete the table below that represents a signaling mechanism in human by writing mechanism 1 till 4. You now need to organize them 1 to 4 or 1 to 5. Basically, it should be 1 till 5, where 1 is the first step of the signaling. Okay, so we have 5 steps over here. So what should be the step number 1 is like the ligand bind to its receptor. So you look for the keyword binds to receptor. So this is step number 1. What happens after that? The receptors, they become activated. As you can see over here, we discussed like when the ligand bind to the receptor gets activated. Look for the keyword receptors activated. So this is number 2. Then number 3, we studied like relay proteins get activated. So these are the relay proteins, they get activated. So relay proteins activated, this is step number 3. Then carrier molecules are sent to the plasma membrane. No. The transduce the signal triggers packaging of the glucose specific carrier molecules. Yes. So this is 4, and the carrier molecules are sent. This is 5. Summarize using the table differences between the signaling of the hydrophilic and hydrophobic molecules. Hydrophilic are the water loving, and hydrophobic are the water hating or water fearing molecules. So hydrophilic is the one which must need a receptor and on cell membrane. So for hydrophobic, receptors are present on the cell membrane, whereas here the receptors are present inside the cell. Receptors are inside the cell. Okay, so we need to look into the 3 types. Okay, so first of all, hydrophilic and hydrophobic, we can go for the receptors. So as we discussed already, hydrophilic, they are the membrane bound, right? And here they are in the inside cell, or you can say in the cytoplasm. What kind of the response comes, like if it is fast or if it is slow? Since it is membrane bound, so it's going to be like very rapid. And since it is rapid, like the nervous system, when the nerves are very rapid, so it's short lived. So like the reaction will be very quick. Hydrophobic, of course, if the above was rapid, so it is going to be slow. It's going to be slow. And then if it is the above was short lived, so it's going to be long lived. Now what are the examples for at least remember, like hydrophobic are steroids, which are lipid soluble, so they can easily cross through the membrane, while like adrenaline is one of the example of the hydrophilic molecules, or ACTH, remember that. Complete the table below to compare signaling of the hydrophilic and hydrophobic molecule. It's the same thing as we did up, but now it's in a different way. And here we are going to talk about like receptors, the same stuff, it's the same stuff, but we will say it in little bit different way. So we will say, for example, receptor. So receptor is for the hydrophilic. We said already here the receptor is membrane bound. It's the membrane bound. And over here it's in the intracellular. Okay, now cellular response, as we said up, this is the same table as a flipped one. So it is rapid, and it is slow, it's short lived, right? And this one is long lived. Okay, and then examples, the same stuff you wrote over here. It's adrenaline or acetylcholine, as I told you, and over here the steroids. So it's the same table but just a flipped one. Albuterol is an agonist of the hydrophilic signaling. Agonist, like they speed up the reaction. So you have studied 2 terms. One term was agonist, and the other term was antagonist. Okay, so remember those terms. Agonist, they are like speeding up the reaction, and antagonists, they are like the stopping the reaction. Albuterol is agonist of the hydrophilic signaling molecule adrenaline. So its receptors are receptors at the target cell are where they will be. So if it is a hydrophilic, you can go up and see hydrophilic is the membrane bound. So of course you can simply choose it's in the membrane bound. But if they change over here that it's hydrophobic, so simply for the hydrophobic you will go that the receptors are in the cytoplasm. Differentiate between agonists and antagonists. Yes, these are the terms we came up now. Which of the following correctly describe the caffeine works shown in the diagram? So you need to see like how the caffeine is working over here. So when there is no caffeine, okay, decreased alertness and increased sleepiness is there when there is no caffeine. But when you add caffeine, so the process change, it's like increased alertness and decreased sleepiness. So you can simply see over here what does the caffeine is doing basically. So caffeine is making the G protein coupled reaction as inactive. So caffeine works as what? Antagonist or agonist, that you have to see. So of course it is making it inactive. So when it is making it inactive, it is antagonist. So look for the words antagonist. So agonist, no. Agonist, no. Antagonist, yes, if it is either C or D, but we need to read it now. Caffeine works as antagonist since the signaling pathway was activated. No, we can see the signaling pathway was inactive, or deactive. So D is the correct answer. Now, the drug albuterol is an agonist to adrenaline signaling because we already said like sorry, I skipped one question. So what is the effect of the caffeine binding to receptors on the brain? So what does the caffeine is doing? So basically caffeine is acting as an antagonist. So again we will see that agonist, no. Antagonist, yes. Agonist, no. So A and C are not answered. We will go for BRD. Caffeine is an antagonist in the heart but agonist in the lungs. No, it cannot work like that way. It should be same, either agonist or antagonist. Caffeine is an antagonist to adenosine receptors, the brain, blocking the agonist. Yes, this blocking the adenosine response. Yes, that's the correct one. Drug albuterol is an agonist to adrenaline signaling because it inhibits, no. It evokes responses similar to those induced by adrenaline. That can be possible. Evokes mean like, for example, it activates or it continues like that. Reduces, no. Decreases, no. So inhibit, reduce and decrease. Look, inhibit, reduce and decrease, they are all synonyms, they mean the same. So these three options cannot be correct answer when you are talking about the agonist. A group of scientists has isolated a protein from the plant. They hypothesize it is an agonist to a particular receptor, on an animal cell. The scientists grow a group of target cells from the animal in a solution that mimics the typical environment of the cell, and they then added the plant protein and measure the response of the animal cell. What outcome would support the hypothesis? What was their hypothesis actually? So they hypothesized that it's an agonist to a particular receptor on the animal cell. Okay. Now, there would be a cellular response, like how it shows you that what's happening. So you need to look for that. If it is binding to a particular receptor and it is an agonist, yes, that can result in cellular response. There should be no cellular response. No, this is antagonist. Okay, and the signal transduction pathway would not progress, again antagonist. The signal transduction pathway would be bypassed, again antagonist. So A is the correct answer. Describe using example how negative feedback helps to maintain homeostasis. As a result of the process shown, there the process is missing over here. The glucose level decreases in the blood. Identify if it is positive or negative. Of course, when the glucose level decreases, it's gonna be what? Negative, right? And explain what will happen if the insulin level in the blood so of course when the glucose level is decreasing, the insulin level will decrease as well, while the glucagon level will increase. So based on the diagram given, you should know that it is a negative feedback mechanism. Describe using I think you can refer this to IPQ. Okay, so refer to the original IPQs. You can find the picture over there and this question over there. Okay, so try to refer to the original IPQs which were provided in 5.1. Describe using examples. The positive feedback is self-reinforcing and amplifies response. Which of the following is correct about the diagram below? So you need to look at the diagram below. Apple on tree ripens. Ripe apple produces ethylene. Ethylene signals the neighboring ripened cells. Neighboring cells produce more ethylene and more of course, it's an example of the positive, because it amplifies more. More mean amplifies, so it's positive feedback. Negative, no. Negative, no. It's an example of positive feedback since the final process stops the process, no. And amplifies, yes. So D is the correct. You always have to look for the keywords to identify the what's happening. Use the diagram below to answer question 49 till 51. And we have here uterus muscle cell, pituitary gland, oxytocin hormone, oxytocin more, and then the oxytocin binding over there. So identify whether the oxytocin hormone is hydrophilic or hydrophobic. Look, oxytocin is binding to who? To the receptors in where? In the cell membrane. So anyone who binds to in the cell membrane is of course hydrophilic. And use two pieces of evidences from the diagram to support your answer. So one piece of evidence I have already told you that it's a hydrophilic. Why? Because it has receptors in the cell membrane, right? What else you can see over here to support? You can see the secondary messengers over here. Like after that, the G protein get activated, and then there is a phospholipase C, which is a secondary messenger, which can act as a secondary messenger. And also the cytoplasmic activity is there. Okay, so cytoplasmic activity is over there, that helps the process to take place. Identify whether the diagram shows a positive or negative feedback. Of course, it's a positive feedback. We know that whenever there is amplification or the muscle contraction is increasing more and more, so it's a positive. It's a positive. So over here, what are the two pieces of evidence? Number one, the membrane receptor. Number two, secondary messenger involved. And number three, it's like cytoplasmic activity can be seen. Cytoplasmic activity or cytoplasmic reactions can be seen. Okay. Now, positive or negative, explain your answer. So it is positive because you can see over here uterus muscle contraction, and then muscle contraction is increasing more and more. So you can say the explanation is like amplification of uterine muscle contraction can be seen. Okay, explain how the pathway shown in the diagram causes labor in human. So of course, the labor in human, like the process baby pushes the uterine wall, causing the stretch. Then the stretching causes the pituitary hormone to release more oxytocin. Then the oxytocin hormone acts on the uterine wall, and continuous contractions lead to the delivery of the baby. So please look for the answer key, okay, for full answer. Describe using, okay, topic change 5.2.2. Describe using visuals and representation different events that occur during the cell cycle. Please make sure you are familiar with the cell cycle to answer such questions. So this is cell cycle. Okay, we have main phases like G1, then we have S, then we have G2. Okay, so we have to see this is G1, then this is S, this is G2, and then this is M. So what happens over here? This completes the cell cycle. But just after the M phase, over here we have the next phase that's called as cytokinesis. Okay, we have cytokinesis. And also remember that the M phase is divided into PMAT. So it is divided into PMAT: prophase, metaphase, anaphase, and telophase. Now remember at least one major event that takes place in each phase. So then you can be going through it. And also just before, if the cell is not dividing before the G1, it's called as G0 phase. The cell stays in the rest. Okay. The diagram is given same as diagram given below. If you look into my diagram up, you can easily label that A is G1, B is S, C is G2, this is P, M, A, T, and this is cytokinesis. So you can do it. And also you can see like this phase, okay, it's given over here, the inner row. This is interphase. This is interphase, okay. DNA replication occurs when in the S phase. So as I told you, DNA replication. So remember one event that as the nuclear envelope breaks down in the early mitosis, no. During mitosis, no. During S phase, yes. In animal cells, but not in plant cells, no. During which phase the cell cycle does mitosis happen? So of course, as I told you, M phase, right? So this is M. And which of the following shows the correct sequence of the cell cycle? Cytokinesis to mitosis? No. S? No. G1, S, G2, M, and S, T, yes, this one. Mitosis to G1 can be possible. To S, to G2, and to cytokinesis. No, until here it is correct. Okay, but not this one. So this is wrong. Use the figure to answer 55 and 56. Which letter represent the stage during which that prepares for division? So in which phase it is preparing for division? That is G2, because after G2 there is M phase. So in M phase the cell will divide. So it is G2, that is represented by the C. Which letter represents the stage during which DNA replication occurs? That's the S phase, and it is B. Okay. Identify the relative time spent to complete each phase in typical human. You can see the time given over here is 1, 9, 1 plus 9. So you can add them all. 1 plus 9 plus 7 plus 3. That's 20 hours in total. So what percentage of the time required to complete the full cell cycle do they typically spend in interphase? So interphase is the sum of G1, S, and G2, right? So that is 9 plus 7 plus 3, and that's 19 hour. So it's 19 out of 20. That's like 95%. Right? So you can go with the answer D. So you need to check the total. So for example, now we are left with this M, right? M. So if M is only 1 hour, so how you can calculate for M? So it is 1 out of 20. So 1 by 20, that will be 5%. So this is how it works. If the total time taken for the cell cycle is 20 to 200 minutes, calculate the time in minutes that the C phase takes place. The diagram for this is in the original IPQs. You need to check the original IPQs, and they said their C phase takes 2.4% of the total. So you can simply say 2.4% of 200. Okay, 2.4% of the 200, you can do like this. So if you do 2.4% of the total, how you can do the answer calculation? I think it's not 2.4, it's 2.8. Yes, that's 2.8. You will see it in the calculations. So 2.8% times 200, so that's like 2.8 divided by 100 times 200. So this will cut. So it's 2.8 times 2, that's 5.6 minutes. Okay, so this is how the answer will come. Okay. Describe using representation different stages of mitosis. As I told you, PMAT: prophase, metaphase, anaphase, telophase. Learn at least 1 point for each phase so you can answer them. Which of the following stages is shown over here? You can see over here the chromatids are split. Here the chromatids are split. So when the chromatids are split, this is anaphase. So in the prophase, basically condensation takes place, condensation, duplication. Metaphase, all chromosomes line up in the center, telophase, like two cells appear, nuclear membrane reappear, two cells appear, and anaphase that you can see over here. The diagram below shows a stage. So just now we said chromosomes line up in center, so now you can say this is metaphase. Brings directly after mitosis, has duplicated chromosomes, is called anaphase. No. Occurs during interphase. No. Has duplicated chromosome. Yes. And begins directly after mitosis. No, because right after mitosis it's G1 and then GS, then G2, and then so on. So the answer is B. Use the diagram below which represents the mitosis stages to complete the table. So there is a table down, but we can do over here. All chromosomes are lined up in the center. This is metaphase. Two cells are formed. You can see the chromatids in the cell. The nuclear envelopment cell is about to divide. This is telophase. Then the chromosomes are not in the center and not away. So this is early metaphase. Okay, then you can see the chromosomes are there and the spindle fibers are moving there. So this is prophase. You can say this is early prophase, and then this is anaphase because the chromosomes are just about to be split. So or this can be prophase or you can say early prophase or prometaphase. Okay, whatever you say, that will be the fine answer for this one. Okay, so I told you all these stages and the description, so look for the answer key for here. 5.3.1, the third, where we will see the regulation of the cell cycle that's brought about by the cyclins and cyclin dependent kinases like CDKs. Okay, so the table below shows 3 types of the cyclins. Actually there are 5 types of the cyclin. There is D and D1 as well. Regulatory proteins of the cell cycle and the phase at which each one has its maximum concentration. The table below shows information that we will use to answer question 62 and 63. Cell cyclin type is A, B, C. A is mostly involved in S phase, B is in M phase, and E is in the G1 phase. Predict one function of the cyclin B during the cell cycle. When the cyclin B is more in the M phase, so what it can do? So M phase is like mainly about mitosis. So basically it's related to start of the mitosis, that's the condensation of chromosomes. So it's mostly involved in condensation of chromosomes, chromatin to form chromosomes. Identify the cyclin that if in low concentration the cell will enter to G1. If you go back, as I told you, the cell goes in G1 when G0, sorry, when the G1 is not taking place. So any cyclin which is responsible for the G1, if it is not available, the cell will go into the G0. So this is cyclin E. Okay, cyclin E. Identify, and now you have to explain your answer. So explaining is quite easy, like if the E is in more concentration, the cell will go through G1 phase, but if it is not there, if it is in low, the cell will stay there in the G1. And now as per the new terminology, if the cell is staying longer in the G1, this stage is turned out to be G0. Use the diagram below to answer 64 till 66. So this is the diagram again, same cell cycle, G1, S, G2 and M phases, and these are different cyclins on the top which are involved in each phase. There are multiple types. Okay, we already discussed about these. Use the data in the graph, determine whether the percentage of the cyclin types are what percentage are necessary for the cell to move from S to G2. So this is S, and to move into G2 you need to look in between these two phases, S to G2. Okay, in between these two phases, how many cyclins are involved. So I can only see one cyclin which is involved over here, right? Cyclin A. So the answer is like 1 out of 5. So 1 out of 5 is 20%. Okay. There are multiples. Okay, same statement. Use the data in the graph to determine which type. Now we need to name which type of the cyclin necessary to move from S to G2. Okay, so here it is S and here it is G2. Again, the same question in a different way. It's only cyclin A. So you can look like this. For example, if we say from G1 to G2, okay, if we say from G1 to G2, then you need this peak, this peak, this peak, and this peak, 4 peaks. So from G1 to G2, you need cyclin D1, D, E, and A. You need all. But over here, for example, they just need from S to G2, so it is cyclin A only, right? Yeah, a combination of which cyclin is most likely responsible for initiating and performing DNA replication in the cell cycle. DNA replication takes place in which phase? S phase. So combination mean like there are two cyclins. So we will see S phase. So this is S phase. Which cyclins help in the completion of the S phase? You need to look into here. So that is this peak and this peak. So these two peaks are very important for the completion of the S phase. So that the S phase completed and then it goes to this one. So it is cyclin E and A, because E is bringing it to the S phase and A is completing the S phase. So both are involved. So the answer is E and A. Explain using visuals and representations how the cell how the cell cycle is regulated in eukaryote cell through checkpoints. So these are the checkpoints like G2, M, and G1 checkpoints. Use diagram to answer this. Which of the following is required to pass the M phase? M phase checkpoint is like to check if all the chromosomes are lined up and they are connected to the spindle fibers, right? So I just told you this one. Accurate completion of DNA replication. This is G2 checkpoint. Adequate number of free nucleotides, no need, not related to checkpoints. The completion of the process of cytokinesis, not related to checkpoint. So this is the M checkpoint. Which type is required? Which of the following is required to pass the G1 checkpoint in the diagram below? G1, as I told you already, G1 is basically related to the accurate and complete DNA replication. No, this is G2. Adequate number of active mitochondria, nothing related. Proper attachment, this is M checkpoint. The cell has reached to sufficient size. Yes, this is G1. With that, we reach to end of the mitosis, cell regulation, cell cycle regulation, and cell signaling. We will continue for the next in another video. Thank you.